Some ideas become so familiar that we stop asking if they're true. They don't feel like theories anymore. They feel like facts. Quiet, settled, unquestioned. Human origins is one of them. For generations, we were taught a story that felt complete. A story with a beginning, a path, and a conclusion. A people formed in one place, scattered by history, and reunited by memory. It was neat. It was comforting. And most importantly, it was rarely challenged. Until DNA forced us to look again. For centuries, historians, theologians, and linguists tried to reconstruct the past using texts, traditions, and ruins. Sacred scriptures were read as chronicles. Ancient maps were treated like snapshots of truth. Family lineages were preserved in words, not molecules. And for a long time, that was enough. But beneath every written record, beneath every preserved ritual, something else was quietly waiting. A biological archive far older than any manuscript. A record written not in ink, but in cells. This story begins not with a person, but with a method. In the late 20th century, a new kind of investigator entered the historical debate. Geneticists, armed not with scrolls or stones, but with sequencing machines and probability models, began to ask questions historians couldn't answer alone. Who were these people, really? Where did they come from, biologically, not symbolically? And how much of the story had been shaped by repetition rather than evidence? At first, the resistance was subtle. Many scholars believed genetics would simply confirm what was already known, that DNA would politely fall in line behind established narratives, that it would validate tradition, not complicate it. But science rarely behaves so obediently. As genetic data accumulated, a pattern began to emerge, not a clean line, but a mosaic, not a single origin, but overlapping histories layered across continents and centuries. The protagonists of this story are not individuals. They are populations. Jewish communities spread across the globe, Ashkenazi in Europe, Sephardi across Iberia and North Africa, Mizrahi in the Middle East, Beta Israel in Ethiopia, and smaller, lesser-known groups in India and China, had lived apart for centuries. They spoke different languages. They absorbed local customs. They survived under wildly different political realities. Yet they shared something intangible, a sense of belonging to the same story. The question was whether that story existed in their DNA, or only in memory. Geneticists approached this problem like detectives revisiting a cold case. Instead of asking what people believed about their origins, they asked what their cells remembered. And DNA, unlike tradition, does not lie politely. The first breakthrough came from an unexpected direction, the Y chromosome. Passed almost unchanged from father to son, the Y chromosome acts like a biological surname, tracing paternal ancestry across thousands of years. When scientists analyzed Y chromosomal markers from Jewish men across Europe, North Africa, and the Middle East, they found something striking. Despite centuries of separation, many shared a common paternal signature, one that pointed not to Europe, but to the Levant, to a region encompassing modern-day Israel, Jordan, and Lebanon, to a time roughly 3,000 years ago. For the first time, biology echoed history. But this confirmation came with a complication, because DNA does not tell one story. It tells many, depending on where you look. While the paternal line suggested continuity, another genetic archive told a far messier tale, mitochondrial DNA. Unlike the Y chromosome, mitochondrial DNA is passed from mothers to their children, tracing maternal ancestry deep into the past. When researchers examined mitochondrial DNA among Ashkenazi Jewish populations, the results challenged expectations. Instead of Middle Eastern origins, many maternal lineages traced back to Europe, particularly Southern Europe, regions like Italy and Greece. This was not a minor discrepancy. It was a structural contradiction. How could a population be simultaneously Middle Eastern and European? The answer was uncomfortable, not because it was shocking, but because it disrupted a story that had grown too familiar. What emerged was not a single migration, but a series of encounters. Jewish men originating from the Levant had moved into Europe over centuries, forming communities, marrying local women, and raising families within a shared cultural framework. The identity remained, the biology diversified, and this was only the beginning. As genetic databases expanded, Researchers began comparing Jewish DNA not only internally, but against neighboring populations. The results revealed shared ancestry with ancient Canaanites, Phoenicians, and Arab groups, peoples who once inhabited the same landscapes and traded, intermarried, and fought long before modern borders existed. History, it turned out, was not composed of isolated peoples moving through empty lands. It was crowded, entangled, human. Wars and empires accelerated this complexity. Assyrian conquests, Babylonian exile, Roman destruction, forced migrations that scattered genetic material across continents like seeds in the wind. Each displacement left traces, not only in chronicles, but in chromosomes. Yet for centuries, academic frameworks resisted this complexity, not out of malice, but inertia. Once an origin story settles into textbooks, it becomes difficult to dislodge. Challenges are seen as noise. Anomalies are explained away, until evidence becomes too consistent to ignore. DNA did not arrive to erase history. It arrived to complicate it. And in doing so, it exposed a deeper truth, that identity is rarely the product of purity or isolation. It is shaped by movement, contact, and survival. This realization forces us to confront an uncomfortable question. How many other origin stories do we accept simply because they feel familiar? And what happens when biology refuses to support them? In the next part of this investigation, we will follow the genetic trail deeper into the mechanisms that created these patterns, the founder effects that reshaped entire populations, and the hidden costs and unexpected benefits of isolation. Because once you start questioning origin stories, you can't stop at just one. Once DNA entered the conversation, history stopped being linear. What had once been described as a clean sequence of events, origin, exile, return, began to fracture into overlapping timelines. Genetics didn't replace historical narratives. It exposed where they were incomplete. To understand how this happened, scientists had to separate ancestry into distinct biological pathways, not metaphorical ones. Literal genetic routes passed down through generations. The investigation followed two parallel trails. The first was paternal. The Y chromosome, inherited almost exclusively from father to son, is remarkably stable over time. Small mutations accumulate slowly, allowing researchers to reconstruct deep ancestral relationships across thousands of years. When geneticists analyzed Y chromosomal markers from Jewish populations across Europe, North Africa, and the Middle East, a clear pattern emerged. Despite long geographic separation, many shared closely related Y haplogroups, clusters of genetic markers that pointed back to the same region. The Levant. This wasn't a vague association. The genetic distance between these paternal lineages and those of neighboring Middle Eastern populations was significantly smaller than with surrounding European groups. In other words, Jewish men in Poland or Morocco were genetically closer, on their paternal line, to populations in the Eastern Mediterranean than to their immediate neighbors. Biology was confirming something historians had long argued, a shared ancient origin in the Near East. But science rarely delivers answers without complications, because ancestry does not travel down a single line. The second trail, maternal ancestry, told a very different story. Mitochondrial DNA, passed from mothers to all their children, follows a separate inheritance path entirely. When researchers turned to mitochondrial markers, especially within Ashkenazi Jewish communities, the results were unexpected. Instead of Middle Eastern origins, many maternal lineages traced back to Europe, Southern Europe, to be precise, Italy, Greece, the Mediterranean basin. This wasn't a marginal finding. A significant portion of Ashkenazi mitochondrial DNA clustered tightly with local European populations, not with Middle Eastern ones. The implication was unavoidable. At some point, Jewish communities in Europe had absorbed a large number of local women whose descendants would carry their mitochondrial DNA forward for centuries. This did not mean forced conversion or a single historical moment. The genetic data suggested something more gradual and human. Small communities, migration, intermarriage, and cultural continuity maintained through tradition rather than biology alone. In simple terms, many Jewish populations carried Middle Eastern fathers and European mothers, a dual ancestry. And that duality explained something historians had struggled with for decades: how a dispersed population could remain culturally cohesive while biologically diverse. Language offered a striking parallel. Yiddish, spoken by Ashkenazi Jews for centuries, is not a pure language. It is a fusion, primarily Germanic in structure, infused with Hebrew religious vocabulary, and influenced by Slavic languages from Eastern Europe. Genetically, the population followed the same pattern: not replacement, not erasure, but layering. As scientists widened their comparisons, the story grew even more complex. Jewish genetic profiles showed consistent overlap with ancient Levantine populations, including groups historically labeled as Canaanites and Phoenicians. These were not distant associations. They reflected shared ancestry rooted in the same geographic corridor thousands of years ago. At the same time, genetic proximity to Arab populations across the Middle East revealed centuries of shared history long before modern ethnic labels solidified. This was uncomfortable for rigid narratives, because it challenged the idea of isolated peoples moving through empty land. The ancient Near East was a genetic crossroads, dense, interconnected, and constantly reshaped by trade, conflict, and migration. And then came empire. Assyrian campaigns displaced entire populations. Babylonian conquests forced elites into exile. Roman destruction scattered communities across the Mediterranean world. Each event acted like a genetic dispersal mechanism, redistributing ancestral material far beyond its original homeland. These were not symbolic exiles. They were biological ones. Genes do not disappear when borders change. They move with people, quietly, persistently, indifferent to ideology. Over time, small founder populations emerged, groups of limited size, carrying only a subset of the original genetic diversity, established communities far from their point of origin. This process amplified certain genetic traits, not because they were advantageous, but because chance favored them. The result was genetic clustering. Distinct Jewish populations began to develop unique genetic signatures, shaped by geography, isolation, and social structure, while still retaining traces of shared ancestry beneath the surface. This explains why Jewish communities can appear genetically close in one analysis and strikingly different in another. It depends on which layer of history you examine. Zoom out far enough, and the connections emerge. Zoom in closely, and the differences dominate. For decades, academic frameworks struggled to reconcile these findings. Some researchers emphasized continuity. Others focused on divergence. Debates hardened into camps. But DNA does not choose sides. It simply accumulates data. And as datasets expanded, thousands, then tens of thousands of samples, a new consensus began to form. Jewish origins were neither a single biological lineage nor a purely constructed identity. They were a product of migration, integration, and memory. A population defined as much by who joined it as by where it began. This realization reframed the entire discussion. Identity was no longer a question of genetic purity. It became a question of persistence. How did a dispersed population maintain coherence across centuries of biological change? And what were the consequences, both biological and cultural, of living in small, tightly knit communities for generations? To answer that, scientists had to look not at origins, but at bottlenecks, at moments when history narrowed the genetic future of entire populations. And nowhere is this more visible than in the case of the Ashkenazi Jews, and a phenomenon known as the founder effect. That is where the investigation turns next. To understand how genetics can shape destiny, scientists look for moments when history narrows options, moments when populations shrink, when survival depends on a few families, when chance, not choice, decides which genes move forward. In population genetics, this phenomenon has a name: the founder effect. It occurs when a small group breaks away from a larger population and establishes a new community. The genetic makeup of that group, incomplete, uneven, accidental, becomes the foundation for generations to come. And few populations illustrate this more clearly than the Ashkenazi Jews. Historical records suggest that medieval Ashkenazi communities began as relatively small groups in Central Europe. Political instability, religious persecution, and economic restrictions kept these communities isolated, socially, geographically, and genetically. Marriage outside the community was rare. Movement was limited. Over centuries, the population grew, but from a narrow genetic base. This created a paradox. Culturally, Ashkenazi Jews were remarkably resilient. Biologically, they were increasingly constrained. As geneticists analyzed Ashkenazi DNA, they noticed something unusual. Certain genetic variants appeared far more frequently than in surrounding European populations, not because they were beneficial, but because they were inherited from a small number of founders. Chance had amplified them. This amplification came with consequences. Some of these variants were linked to inherited disorders, Tay-Sachs disease, Gaucher disease, familial dysautonomia, and others. Conditions that were rare elsewhere appeared at significantly higher rates within Ashkenazi communities. For generations, these diseases were misunderstood, sometimes attributed to superstition, sometimes treated as tragic coincidences. But genetics offered a clearer explanation. When a small population expands rapidly from limited ancestors, rare mutations can become common, not through selection, but through inheritance. This realization reframed centuries of suffering. The same historical isolation that preserved cultural identity had also concentrated genetic risk. But this is where the story takes an unexpected turn, because the founder effect did not only create vulnerability, it also created clarity. The genetic consistency within Ashkenazi populations made them uniquely valuable to modern medicine. Patterns that would be difficult to detect in more genetically diverse populations became visible. In a sense, history had created a natural laboratory. Researchers could trace disease-causing mutations with extraordinary precision. Carrier screening programs emerged. Early diagnosis improved outcomes. Genetic counseling became part of community healthcare. What was once a burden became a tool. This transformation, from isolation to insight, highlights a deeper truth about genetics. It does not assign meaning. Humans do. The same DNA that carried risk also carried information, and the same genetic bottleneck that caused suffering ultimately contributed to scientific progress that now benefits people far beyond the Ashkenazi community. But the founder effect was not only medical, it shaped identity. Genetic studies revealed that while Ashkenazi Jews showed Middle Eastern paternal ancestry and European maternal ancestry, centuries of isolation had blended these lines into a distinct genetic signature, neither purely Levantine nor European. A third pattern emerged. This pattern mirrored culture. Ashkenazi identity was not a preserved relic from the ancient world. It was something built through adaptation, memory, and continuity under pressure. Yiddish, once again, offers a powerful analogy. Like Ashkenazi DNA, Yiddish did not replace its components. It fused them. Germanic grammar carried Hebrew religious life, infused with Slavic rhythms. A living system shaped by constraint. The same forces that limited genetic diversity also intensified cultural cohesion. Shared rituals, education, and social structures became survival mechanisms, binding communities together when external forces pushed them apart. Yet this cohesion came at a cost. Every bottleneck leaves scars. Genetic data reveals periods where diversity sharply declined, moments when population size dropped dramatically due to persecution, expulsion, or famine. Each collapse narrowed the genetic future of the community. And yet, the community endured. This endurance complicates simplistic narratives of identity. It challenges the idea that genetics alone defines belonging. Ashkenazi Jews were not united by biological purity. They were united by shared memory, shared risk, and shared survival. And this realization extends beyond one group. Founder effects appear across human history, among island populations, religious isolates, and migrant communities. They reveal how fragile and contingent ancestry truly is. Identity emerges not from ideal conditions, but from constraint. This understanding forces a broader question. If Ashkenazi genetic identity is the product of historical bottlenecks, intermarriage, and chance, what does that say about identity elsewhere? And what happens when DNA contradicts the stories people tell about themselves? To explore this, geneticists turn to communities that did not fit expected patterns at all. Groups whose cultural identity persisted even when their DNA pointed elsewhere. Communities that tested the limits of biological definitions. In Ethiopia, in India, in China, places where Jewish identity survived without clear genetic continuity. These cases forced the investigation beyond ancestry, toward meaning. And that is where the story moves next. By the time genetics reached beyond Europe and the Middle East, the investigation had already dismantled one illusion. That identity could be reduced to blood. What researchers found next would challenge an even deeper assumption, that biology alone can determine who belongs to a people. Because not all Jewish communities fit the genetic pattern. Some didn't even come close. In Ethiopia, the Beta Israel community had preserved Jewish rituals for centuries. Sabbath observance, dietary laws, religious texts adapted to local tradition. Their identity was not theoretical. It was lived, transmitted, defended. But when geneticists analyzed their DNA, the results were surprising. Most Beta Israel genetic markers aligned closely with neighboring East African populations, not with Levantine or European Jewish groups. The biological connection to ancient Israelite populations was weak or absent. By genetic standard alone, they should not have existed as a Jewish group. And yet they did. The same contradiction appeared thousands of kilometers away. In India, small Jewish communities, such as the B'nai Israel and Cochin Jews, traced their origins to shipwreck legends, trade routes, and oral history rather than documented mass migration. Genetic studies revealed partial Middle Eastern ancestry in some cases, but also extensive local integration. In China, the Kaifeng Jews maintained Jewish practices for nearly a millennium, long after losing contact with the wider Jewish world. Their DNA, however, reflected deep integration into the local population. These communities posed a problem that genetics alone could not solve. Because identity, it turned out, is not inherited the way genes are. It is transmitted differently, through ritual, through memory, through shared narratives repeated across generations. DNA can tell us where people came from biologically. It cannot tell us why they stayed who they were. This distinction forces a reevaluation of what origin stories are meant to do. For centuries, origin myths served as anchors. They provided coherence, legitimacy, and belonging. But when they become rigid, when they resist revision, they turn into prisons. Academic inertia thrives on familiar frameworks. Once a story is accepted, challenging it feels destabilizing. New evidence is treated as a threat rather than an opportunity. Genetics disrupted that comfort, not by replacing history, but by exposing its blind spots. It showed that populations are not static units moving cleanly through time. They are dynamic systems shaped by migration, conversion, intermarriage, survival, and choice. Jewish identity emerged not as a single genetic lineage, but as a braided history. A Levantine core intertwined with European, North African, Middle Eastern, African, and Asian strands. Some biological, some cultural, some remembered, some forgotten, and yet coherent. This coherence does not come from purity. It comes from persistence. Across exile and return, persecution and protection, destruction and rebuilding, Jewish communities maintained a shared sense of belonging, even as their genetic makeup shifted. This challenges a deeply ingrained assumption that ancestry determines identity. In reality, ancestry explains structure, not meaning. Genes explain probabilities, not purpose. They can tell us who shared ancestors. They cannot tell us who shared destiny. This realization extends far beyond Jewish history. Every human population carries similar complexity. Migration reshapes ancestry. Isolation amplifies traits. Identity adapts. What makes this case unique is not the genetics, but the way memory endured despite biological change. The story DNA tells is not one of contradiction. It is one of continuity through transformation. And this brings us back to the question that opened this investigation. Why do some origin stories go unquestioned? Because they feel stable. Because they simplify complexity. Because they offer certainty in a world shaped by chance. But certainty is not the same as truth. DNA did not arrive to invalidate history. It arrived to remind us that history is unfinished, constantly revised by new tools, new data, and new perspectives. What we inherit biologically is only part of the story. What we inherit culturally, stories, rituals, values, shapes how those genes matter. In the end, human origins are not a fixed point in the past. They are a process, written not only in chromosomes, but in choices, who people marry, where they settle, what they remember, and what they refuse to forget. The danger was never that we believed the wrong origin story. The danger was that we stopped questioning it. Because once we stop asking where we come from, we stop understanding who we are. And DNA, silent for millennia, was never meant to replace our stories. It was meant to wake us up,